Welcome to part 3 of our discussion on the endomembrane system. Last meeting, we were able to talk about the different components of the endomembrane system, and it includes the endoplasmic reticulum, the Golgi complex. Today, we are going to talk about the lysosomes as well as the peroxisomes. We will also talk about the transport of materials that are within coated vesicles. As a recap, recall that one of the functions of the smooth endoplasmic reticulum is to store glucose in the form of glycogen, and that this glycogen, before it can be transported out of the cell, would have to be dephosphorylated via the action of the glucose 6 phosphatase. The glucose 6 phosphatase will remove the inorganic phosphate from glucose 6 phosphate, and it allows now glucose to be transported via glucose transporters. We also learned that initial modification of protein products happen in the endoplasmic reticulum, whereby biosynthesis of core oligosaccharides, processing of these core oligosaccharides, and addition of such would happen in the ER. Furthermore, the ER also has an additional function of quality check, so that misfolded proteins would not be able to proceed to the next compartment, which happens to be the Golgi apparatus or the Golgi network. Now, the Golgi network is the site where final modification and eventually packaging of the gene product, in this case the protein, would take place. Membrane-bound and soluble proteins synthesized in the rough ER must be directed to a variety of intracellular locations, including the ER itself, the Golgi apparatus, endosomes, and lysosomes. Moreover, once a protein reaches an organelle where it is to remain, there must be a mechanism for preventing it from leaving. Other groups of proteins synthesized in the rough ER are destined for incorporation into the plasma membrane or for release to the outside of the cell. Therefore, each protein contains a specific tag, targeting the protein to a transport vesicle that will carry material from one specific cellular location to another. Depending on the protein and its destination, the tag may be a short amino acid sequence, an oligosaccharide side chain, it could be a hydrophobic domain, or maybe some other structural feature. Tags may also be involved in excluding material from certain vesicles. Membrane lipids may also be tagged to help vesicles reach their proper destinations. This tag can be one or more phosphate groups attached to positions 3, 4, or maybe and/or position 5 of a membrane phosphatidylinositol molecule by specific kinase. For example, a functional phosphatidylinositol 3 kinase is required for proper sorting of vesicles to the vacuole in the yeast. In mammalian cells, inhibition of inositol kinases disrupts vesicle trafficking to the lysosome. Aside from specific tags on some proteins, the length and degree of saturation of certain membrane lipids have also been shown to be important in vesicle trafficking. An overview of the trafficking involving the ER and the Golgi complex will be presented in the next slide. Sorting of proteins begin in the endoplasmic reticulum and early compartments of the Golgi stack, which would contain mechanisms for retrieving or retaining compartment-specific proteins. This important step preserves the compartment-specific functions needed to maintain the integrity of glycosylation and processing pathways. The final sorting of material that will leave the Golgi complex occurs in the TGN where lipids and proteins are selectively packaged into distinct populations of transport vesicles that are destined for different locations in the cell. In some cells, the Golgi complex is also involved in the processing of proteins that enter the cell by endocytosis. This figure shows the trafficking through the endomembrane system. Vesicles that carry lipids and proteins along several routes from the ER through the Golgi complex to various destinations, including the secretory vesicles, endosomes, and the lysosomes. In number 1, here, colored blue, as the ribosomes attached to the rough endoplasmic reticulum synthesizes proteins, the proteins enter the ER lumen, where initial glycosylation steps occur. Glycosylation therefore occurs co-translationally. In number 2, transition vesicles carry glycosylated proteins to the CGN. Finally, number 3, anterograde transport happens, where lipids and proteins move through the cisternae of the Golgi stack via shuttle vesicles, and cisternae mature as new enzymes are added via vesicles. This follows the model for the Golgi apparatus maturation. In number 4, at the TGN, some vesicles will bud off to form the secretory vesicles, which will now move towards the plasma membrane and thereby release its contents via exocytosis. On the other hand, here, other vesicles would bud off from the TGN to form endosomes. These endosomes will in turn help to make lysosomes. In the next step, the proteins and other materials are taken into the cells by means of endocytosis, forming endocytic vesicles that fuse with early endosomes here. The early endosomes, containing material for digestion, eventually mature so that it develops to become late endosomes, and then into lysosomes. This is following the retrograde transport. Retrograde traffic returns the compartment-specific proteins to earlier compartments where it is destined or where it is needed. The protein composition required in the endoplasmic reticulum is maintained both by preventing some proteins from escaping when vesicles would bud off from the ER membrane, and also by retrieving some proteins that have left the ER and reached the CGN. It is not entirely clear, however, how the proteins that never leave the ER are retained. But one theory proposes that they form extensive complexes that are physically excluded from vesicles that bud off from the ER. Several proteins that are localized in the ER may also contain tripeptide sequence, the RXR, otherwise arginine X arginine, where X is any amino acid. This appears to promote retention in the ER. This retention tag is also found in some multi-subunit proteins that are destined for the plasma membrane. The N-methyl D-aspartate, or the NMDA receptor, which is important in neurotransmission in mammalian brain, contains such a tag. But why, you may ask, does this plasma membrane protein have an ER retention signal? It is actually thought that the tripeptide causes the individual subunits of NMDA to be retained in the ER until assembly of the multi-subunit complex in the ER is completed. Proper assembly masks the RXR sequence, allowing the assembled complex to leave the ER. Of course, other evidence suggests that phosphorylation of this protein near the RXR sequence may promote ER release. Retrieval by retrograde transport of vesicles from CGN to the ER is better understood than retention. Many soluble ER-specific proteins would contain retrieval tags that may bind to specific transmembrane receptors facing the Golgi complex lumen. The tags are short C-terminal amino acid sequences such as KDEL, which is lysine, aspartame, glutamine, and leucine, or maybe KKXX, where X can be any amino acids in mammals, and HDEL, histidine, aspartame, glutamine, and leucine in these cells. When a protein containing such a tag binds to its receptors, the receptor undergoes a conformational change. The receptor and ligand complex is then packaged into transport vesicles for return to the ER. Evidence for the importance of retrieval tags comes from experiments involving artificial chimeric proteins, also known as the fusion proteins. These are made by joining DNA sequences encoding two polypeptide segments from different proteins, allowing a hybrid polypeptide to be produced from joined DNAs. Proteins normally secreted from the cells were altered in this manner to include amino acids representing an ER retrieval tag, and these proteins were subsequently found in the ER rather than being secreted. Interestingly, some of these chimeric proteins found in the ER have undergone partial processing by enzymes that are found only in the Golgi complex. This finding indicates that the suspected tag did not simply prevent the escape of proteins from the ER, but actively promoted the retrieval of ER-specific proteins from the Golgi complex. Like resident proteins required in the ER, some precedent proteins are also needed in the Golgi complex, in that these proteins have retention or retrieval tags. In the Golgi complex, just like in the ER, the formation of large complexes are excluded from transport and may play a role in maintaining the protein composition of the Golgi complex. In addition, distinctly different mechanism is also likely at work in the Golgi complex, whereby a mechanism involving hydrophobic regions of Golgi proteins. All known Golgi-specific proteins are integral membrane proteins that may have one or more hydrophobic membrane-spanning domains anchoring them to the Golgi membranes. The length of the hydrophobic domains may determine which cisternae of the Golgi complex each membrane-bound protein is incorporated as it moves through the organelle. Now remember, the thickness of the cellular membranes increases progressively from the ER going to the plasma membrane. Among the Golgi-specific proteins, there is a corresponding increase in the length of hydrophobic membrane-spanning domains going from the CGN up to the TGN. Such proteins tend to move from compartment to compartment until the thickness of the membrane exceeds the length of their membrane-spanning domains, thereby preventing them to migrate. The model of lysosomal enzyme targeting came from studies of the human genetic disorder called eye cell disease. Cultured fibroblast cells from patients with eye cell disease synthesize all of the expected lysosomal enzymes but then release most of the soluble proteins to the extracellular medium instead of incorporating them into the lysosomes. In this figure, you will see the targeting of soluble enzymes to endosomes and lysosomes using a mannose-6-phosphate tag. In step number 1, in the ER, the soluble lysosomal enzymes undergo N-glycosylation followed by removal of glucose and mannose units. In step 2, within the Golgi complex, mannose residues on the lysosomal enzymes are phosphorylated by two enzymes. The first will add N-acetylglucosamine 1-phosphate to carbon number 6 of mannose. The second enzyme will eventually remove that N-acetylglucosamine part. Therefore, it will only leave the phosphorylated mannose residue. In step number 3, the tag lysosomal enzyme will now bind to mannose-6-phosphate receptors in the TGN and will now be packaged into coated transport vesicles that convey the enzymes to a late endosome. In step number 4, the acidity of the late endosomal lumen causes the enzymes to dissociate from its receptors, so that finally in step 5, the receptors are recycled in the vesicles that return to the TGN. Note that the late endosome either matures to form a lysosome or it will transfer its contents to an active lysosome. This brings us to secretory pathway transport molecules to the exterior of the cell. And basically, there are two methods of transporting materials across the plasma membrane. And this would be exocytosis, which is the process by which secretory vesicles release their contents outside the cell, and the opposite of which would be endocytosis, which is the process by which the cells internalize external materials. Endocytosis can either be pinocytosis if the materials that enter the cell are very fine, and it could be phagocytosis if materials that enter are very large molecules. Secretory pathways would transport molecules going to the exterior of the cells with the help of secretory vesicles and secretory granules. And experiments using electron microscopy as well as autoradiography would allow us to see the movement of proteins through the secretory pathways. The proteins can be radioactively labeled briefly, and after a few minutes, like 3 minutes maybe, the proteins could be visualized and primarily seen moving through the rough ER. And a few minutes later, the proteins will begin to appear in the Golgi complex. This was such experiment that was conducted as early as 1967 by James Jamieson and George Palade using the secretory cells from guinea pig pancreatic slices. In this figure here, you will see that the pathway of newly synthesized proteins through a cell was traced using radioactively labeled amino acid, and after washing away unincorporated amino acid, microscopic autoradiography was used in order to determine the location of the radioactively labeled proteins in the pancreatic secretory cells at different time points after injection was made. In figure A, you will see that a few minutes after injection, most of the labeled proteins is still found in the rough ER where it has just been synthesized. In figure B, after 7 minutes, most of the new labeled proteins has moved into the adjacent Golgi complexes as is indicated by the arrows. Eventually, after 37 minutes, the proteins was detected in the vesicles budding from the Golgi and named as condensing vacuoles by the researchers. Meanwhile, in Figure D, after 117 minutes, labeled proteins began to accumulate in dense zymogen granules and vesicles that discharge secretory proteins going to the exterior of the cell. Some of the radioactively labeled proteins was observed in the exterior lumen adjacent to the cell, demonstrating that some of the secretory granules that had released their contents from the cell. Based on this classic experiment, numerous similar studies were conducted. The secretory pathways that are shown in this figure are now better understood. We now can distinguish two different modes of secretion by eukaryotic cells. One is constitutive secretion, and the other is regulated secretion. Let's direct our attention to constitutive secretion. After budding from the TGN, some secretory vesicles will move directly to the cell surface, where they will immediately fuse with the plasma membrane and eventually release their contents by exocytosis. This is an unregulated process. It is continuous and it is independent of specific extracellular signals. Example of this would be the continuous release of mucose by cells that line your intestine. Constitutive secretion was once assumed to be a default pathway for proteins synthesized by the rough ER. According to this model, all proteins that are destined to remain in the endomembrane system must have a tag that diverts them from constitutive secretion. Otherwise, they will move through the endomembrane system and will be released outside the cell by default. Support of this model came from studies in which removal of the K-del retrieval tags on resident ER proteins led to secretion of the modified protein. However, there are more recent evidence that suggests that a variety of short amino acid tags may identify specific proteins for constitutive secretion. While vesicles containing constitutively secreted proteins move continuously and directly from the TGN to the plasma membrane, secretory vesicles involved in regulated secretion accumulate in the cell and then it fuses with the plasma membrane only in response to a specific extracellular signal. A very good example of this would be the release of neurotransmitters. Another example of regulated secretion would be the release of insulin from pancreatic beta cells in response to glucose, and the release of zymogens, which is an inactive precursor of hydrolytic enzymes, from the pancreatic acinar cells in response to calcium or endocrine hormones. Regulated secretory vesicles form by budding from the TGN as immature secretory vesicles. This will undergo subsequent maturation. Maturation of secretory proteins involves concentration of the proteins, which we call as condensation. And this is frequently also a means of proteolytic processing. The mature secretory vesicles will then move close to the site of secretion. However, it remains near the plasma membrane until there is a hormone or a chemical signal that will trigger its release by means of fusion with that of the plasma membrane. Current evidence suggests that high concentrations of secretory proteins in secretory granules promote the formation of large protein aggregates that exclude non-secretory proteins. This could occur in the TGN, where only aggregates would be packaged in vesicles destined for secretory granules, or it could occur in secretory granule itself. Now, the pH of the TGN and the secretory granule lumens may serve as a trigger favoring the aggregation as the material would leave the TGN. The soluble proteins that do not become part of an aggregate in the TGN or a secretory granule would be carried by transport vesicles going to other locations. This figure here shows the zymogen granules. It's an electron micrograph of acinar cells from the exocrine pancreas of a rat. It illustrates the zymogen granules designated as ZG, which contains enzymes that are destined for secretion. They are usually concentrated in the region of the cell between the Golgi complex from which they arise and the portion of the plasma membrane bordering the acinar lumen into which the zymogen granules will then be discharged. In many cases, exocytosis of specific proteins is limited to a specific surface of the cell. For example, the secretory cells that line your intestine release digestive enzymes only on the side of the cell that faces the interior of the intestine. This is what you call as polarized secretion. This can also be seen in nerve cells, which would secrete neurotransmitter molecules only at junctions with other nerve cells. Proteins destined for polarized secretion, as well as lipids and protein components of two different membrane layers, are sorted into vesicles that bind to localized recognition sites on subdomains of the plasma membrane. There are two methods of transporting materials across the plasma membrane. First is exocytosis, second is endocytosis. We will consider exocytosis first. In exocytosis, proteins in a vesicle are released to the exterior of the cell as the membrane fuses with the plasma membrane. A variety of proteins are exported both from animals and plant cells by means of exocytosis. Example, animal cells secrete peptide and protein hormones, mucus, milk, proteins, and digestive enzymes via exocytosis. The process of exocytosis is illustrated in this figure. The vesicles containing cellular products destined for secretion will move towards the cell surface, as shown in step 1, where the membrane of the vesicle fuses with the plasma membrane in step 2. Fusion with the plasma membrane will discharge the vesicle contents towards the exterior, as shown in step 3. In the process, the membrane of the vesicle becomes integrated into the plasma membrane, with the inner surface of the vesicle becoming the outer surface of the plasma membrane. Thus, glycoproteins and glycolipids that were originally formed in the ER and Golgi lumen will now face the extracellular space. The mechanism underlying the movement of exocytic vesicles to the cell surface is not really that clear. What we know is that there is the involvement of microtubules in vesicle movement. For example, in some cells, the vesicles appear to move from the Golgi complex going towards the plasma membrane, following the tracks that are made by microtubules that are oriented in a parallel manner directed towards the vesicle movement. Furthermore, vesicle movement stops when the cells are treated with colchicine, which is a known plant alkaloid that prevents microtubule assembly. The fusion of regulated secretory vesicles with the plasma membrane is usually triggered by an extracellular signal. In most cases, the signal could be a hormone or a neurotransmitter that will bind the specific receptor on the cell surface and will trigger the release or the synthesis of a second messenger within the cell. Now, during regulated secretion, a transient elevation of the intracellular concentration of calcium ions would often appear to be an essential step in the signal cascade leading from the receptor on the cell surface to the exocytosis process. Let us now direct our attention to endocytosis. Most eukaryotic cells would carry out one or more forms of endocytosis for uptake of extracellular materials. In this figure, a small segment of the plasma membrane is shown to progressively transport materials, whereby, in step 1, a small segment of the plasma membrane progressively folds inward, and then it pinches off to form an endocytic vesicle containing the ingested substances or particles in steps 2 to 4. Endocytosis is important for several cellular processes, including ingestion of essential nutrients by some unicellular organisms, and also the defense against microorganisms by our very own white blood cells. When it comes to membrane flow, exocytosis and endocytosis have clearly very opposite effects, where exocytosis will add lipids and proteins to the plasma membranes, endocytosis, on the other hand, removes them. Thus, the steady-state composition of the plasma membrane is a consequence of a balance from both exocytosis and also endocytosis. Through endocytosis and retrograde transport, the cell can recycle and reuse molecules deposited in the plasma membrane by secretory vesicles during exocytosis. Earlier, we mentioned that there are two types of endocytosis. One would be phagocytosis and the other one where liquids or small particles are taken up, which we call as pinocytosis. A very good example of phagocytosis would be the function of the white blood cells like neutrophils and macrophages, as they would engulf and eventually digest foreign materials that are found in the bloodstream or in injured tissues. In the case of amoeba, the amoeba are capable of forming pseudopods, which are folds of the membrane that would surround the object, engulf it, and eventually form a phagosome. The phagosome will eventually fuse with a late endosome, which may mature to become a lysosome, whereby the lysosome eventually will digest the engulfed material. So this diagram shows the process of phagocytosis via the formation of a pseudopod. Let's talk about another type of endocytosis, which is receptor-mediated endocytosis. Cells can acquire certain soluble and suspended materials by a process called receptor-mediated endocytosis, which is otherwise known as clathrin-dependent endocytosis. For this process, cells use specific receptors that are found on the outer surface of the plasma membrane. This is a primary mechanism for the specific internalization of most macromolecules by eukaryotic cells. Depending on the cell type, mammalian cells can ingest hormones, growth factors, enzymes, serum proteins, cholesterol, antibodies, iron, and even some viruses and bacterial toxins via receptor-mediated endocytosis. The discovery of receptor-mediated endocytosis and its role in the internalization of low-density lipoproteins is actually highlighted in one of the boxes in your reference book. Receptor-mediated endocytosis of low-density lipoproteins carries cholesterol into mammalian cells. An interest in familial hypercholesterolemia, which is a hereditary predisposition to high blood cholesterol levels, and therefore to atherosclerosis and heart disease, was led by Michael Brown and Joseph Goldstein to the discovery of receptor-mediated endocytosis, for which they both share a Nobel Prize in 1986. Receptor-mediated endocytosis is illustrated in this figure. The process begins with the binding of specific molecules, we refer to as the ligands, to their receptors. Specific ligand-binding proteins found on the outer surface of the plasma membrane, as seen in step 1. As the receptor-ligand complexes diffuse laterally in the membrane, they encounter specialized membrane regions called coated pits that serves as site for the collection and internalization of these complexes in step 2. In a typical mammalian cell, coated pits would occupy about 20% of the total surface area of the plasma membrane. Accumulation of receptor-ligand complexes within the coated pits triggers accumulation of additional proteins on the inner surface of the plasma membrane. These additional proteins may include adaptor proteins, clathrin, and dynamin, which are required for promoting membrane curvature as well as invagination of the pit, shown in step 3. The invagination continues until the pit pinches off from the plasma membrane, forming a coated vesicle in step 4. The clathrin coat is released, leaving an uncoated vesicle in step 5. The coat proteins and dynamin are then recycled to the plasma membrane, where they become available for forming new vesicles, while uncoated vesicle is free to fuse with an early endosome, shown in step number 6. The speed and scope of receptor-mediated endocytosis is actually impressive. Imagine a coated pit usually invaginates within a minute or so of being formed, and up to 2,500 such coated pits invaginate per minute in a cultured fibroblast cells. So, this figure here shows the progressive formation of a coated vesicle from a coated pit as particles of yolk proteins are taken up by the maturing oocyte of a chicken. Here you will see yolk particles accumulate in a coated pit in step 1, where a shallow invagination of the plasma membrane with a clathrin coat found on its inner surface. In number 2, a deeper coated pit forms as more clathrin is added, forcing the membrane to form a curve and trapping additional free particles of yolk inside. In step 3, as additional curvature eventually leads to the formation of a coated vesicle, this coated vesicle will eventually bud from the plasma membrane, and a complete coated vesicle is just formed just below the plasma membrane in step 5 with an intact clathrin coat. There are several variations of receptor-mediated endocytosis. The epidermal growth factor, or EGF, which stimulates the division of epithelial cells, undergoes endocytosis by a mechanism similar to what was shown earlier. Endocytosis plays a very important role in cell signaling. As EGF receptors are internalized, the cell becomes less responsive to EGF. This process is desensitization. Deficiencies in desensitization caused by defective endocytosis can lead to overstimulation by EGF, resulting in excessive cell growth, cell division, and possible tumor formation. In another variation of receptor-mediated endocytosis, receptors are concentrated in coated bits independent of formation of receptor-ligand complexes, and binding of the ligands to the receptor simply triggers its internalization. In yet another variation, the receptors are not only constitutively concentrated, but they are also constitutively internalized, regardless of whether the ligands have bound to the receptors. Example of this would be the LDL receptors that are described in the box 12b in your book. So what happens after internalization? Uncoated vesicles will eventually fuse with vesicles budding from the TGN, forming now the early endosomes. These early endosomes are the sites for sorting and recycling of materials that are brought into the cells. Eventually, it could mature into a late endosome, or it could mature eventually forming lysosomes. Recycling of plasma membrane receptor molecules is facilitated by acidification of the early endosome. The interior of an endocytic vesicle would have a pH of around 7, while the interior of an early endosome has a pH of 5.9 to 6.5. The lower pH is maintained by the presence of an ATP-dependent proton pump found in the endosomal membranes. The slightly acidic environment of the early endosome will eventually decrease the affinity of most receptor-ligand complexes, thereby freeing the receptors to be recycled to the plasma membrane while newly ingested materials is diverted to other locations. Sorting receptors and ligands is not always as simple as sending the receptors to the plasma membrane and then retaining the ligands in the endosomes. There are three alternative fates for ligand-receptor complexes. First is some receptor-ligand complexes, like say the epidermal growth factor and its receptor, are carried to a lysosome for degradation. Second, others are carried to the TGN, where they enter a variety of pathways transporting material throughout the endomembrane system. And third, the receptor-ligand complexes can also travel by transport vesicles to a different region of the plasma membrane, where they are secreted as a part of a process called transcytosis. This pathway accommodates the transfer of extracellular material from one side of the cell, where endocytosis occurs, through the cytoplasm to the opposite side, where exocytosis happens. For example, immunoglobulins are transported across the epithelial cells from the maternal blood to fetal blood by means of transcytosis. The lysosome is an organelle of the endomembrane system that contains digestive enzymes capable of degrading all the major classes of biological macromolecules: lipids, carbohydrates, nucleic acids, and proteins.
These hydrolytic enzymes degrade extracellular materials brought into the cell by endocytosis and digest intracellular structures and macromolecules that are damaged or no longer needed. Lysosomes were discovered in the early 1950s by Christian de Duve and his colleagues. Differential centrifugation led the researchers to realize that an acid phosphatase, initially thought to be located in the mitochondrion, was in fact associated with a class of particles that had never been reported before. Along with the acid phosphatase, the new organelle contained several other hydrolytic enzymes, including beta-glucuronidase, a deoxyribonuclease, a ribonuclease, and protease. Because of its apparent role in cellular lysis, they call this newly discovered organelle a lysosome. Only after the lysosome's existence had been predicted, its properties described, and its enzyme content specified, was the organelle actually observed by electron microscopy and recognized as a normal constituent of most animal cells. Final confirmation came from cytochemical staining reactions that are capable of localizing the acid phosphatase and other lysosomal enzymes to specific structures that can be seen only by electron microscopy. Lysosomes vary in size and shape, but they are usually around 0.5 micrometer in diameter and is bound by a single membrane. The luminal side of the membrane is coated by glycoproteins, which actually protects the lysosome from being degraded. This figure shows how the lysosome is labeled using cytochemical localization of acid phosphatase that is found in the lysosome. Here, the acid phosphatase was labeled, and eventually labeling shows the deposits of electron-dense lead phosphate inside the lysosomes. Lysosomes are able to maintain an acidic environment of around 4 to 5 inside. ATP-dependent proton pumps in the membrane are responsible for this acidic environment, and there are numerous enzymes inside the lysosomes, which are acid hydrolases in nature. The enzymes found in the lysosomes are synthesized by the ribosomes found on the rough ER, which would eventually transport these products into the lysosomes. They are delivered from the TGN going to endosomes in transport vesicles. Over time, the endosomes will mature into late endosomes, and once all of the enzymes are present, it eventually becomes a lysosome. The final step in lysosome development involves the activation of acid hydrolases. This occurs as the internal environment becomes even more acidic. And this is possible because of the pumping of protons or through fusion of an existing lysosome. Lysosomes contain substances that originated outside the cell are called heterophagic lysosomes, and those whose materials originated inside the cell are called autophagic lysosomes. The degradation of external materials that are brought into the cell is possible by means of phagocytosis or by receptor-mediated endocytosis. In the meantime, the phagocytic vacuoles become lysosomes by fusion with endosomes that later develop or fuse with a lysosome. Eventually, materials that are not digested remains in a lysosome, and the lysosome becomes a residual body after digestion ceases. When cells release the contents by exocytosis, in others, accumulation of debris may continue and may play a role in cellular. Another important task for lysosomes is the breakdown of cellular structures and components that are damaged or no longer needed. Most cellular organelles are in a state of dynamic flux, with new organelles continuously being synthesized while old organelles are destroyed. The digestion of old or unwanted organelles of other cell structure is called autophagy. Autophagy is illustrated in the next figure. There are two types of autophagy. One would be macrophagy and the other one would be microphagy. This figure shows early and late stages of autophagic digestion in rat liver cell. At the top, an autophagic vacuole is formed as a mitochondrion is sequestered by a membrane derived from the ER. At the bottom are several autophagic vacuoles containing remnants of the mitochondria. This is taken from a transmission electron microscope. Take note that most old organelles are eliminated through autophagic digestion. Autophagy occurs at varying rates in most cells under most conditions, but it is especially prominent in red blood cell development. As a red blood cell matures, virtually all of the intracellular content is destroyed, including all of the mitochondria. This destruction is accomplished by autophagic digestion. And so a marked increase in autophagy is also noted in cells stressed by starvation. Presumably, the process represents a desperate attempt by the cell to continue providing for its energy needs, even if it has to consume its own structures to do so. It has been suspected that some cancer cells may be deficient in autophagy, but recent work suggests a direct link between autophagy and cancer. The human version of a gene required for autophagy in yeast cells has been shown to be frequently deleted in human breast and ovarian tumors. Knocking out the function of this gene in a mice caused a decrease in autophagy and an increase in the number of breast and lung tumor. However, some oncogenes that promote cancer suppresses autophagy during tumor development. And this only tells us that more research is needed to understand the role of autophagy to cancer development. While most digestive processes involving lysosomal enzymes would occur intracellularly, in some cases, lysosomes discharge their enzymes to the outside of the cell by exocytosis, resulting in an extracellular digestion. One example of extracellular digestion occurs during fertilization of animal eggs. The head of the sperm releases lysosomal enzymes capable of degrading chemical barriers that would otherwise keep the sperm from penetrating the egg surface. Meanwhile, certain inflammatory diseases such as rheumatoid arthritis may result from the inadvertent release of lysosomal enzymes by white blood cells in the joints, thereby damaging the joint tissue. The essential role of lysosomes in the recycling of cellular components is clearly seen in disorders caused by deficiencies of specific lysosomal proteins. And over 40 such lysosomal storage diseases are known to us. Each are characterized by harmful accumulation of specific substances, usually polysaccharides or lipids. In most cases, the substances accumulate because digestive enzymes are defective, or if not defective, it is missing. But they sometimes accumulate because the proteins that transport degradation products from the lysosomal lumen to the cytosol may be defective. Whatever the case may be, the cells in which material accumulates are severely impaired or destroyed. These are just some examples of lysosomal storage diseases. The first storage disease to be understood was type 2 glycogenosis, in which Young children would accumulate excessive amounts of glycogen in the liver, the heart, and skeletal muscles, and eventually they die at an early age. The problem turned out to be a defective form of lysosomal enzyme, the alpha-1,4-glycosidase, which catalyzes glycogen hydrolysis in normal cells. Although glycogen metabolism occurs predominantly in the cytosol, a small amount of glycogen can enter the lysosome through autophagy and will accumulate to a damaging level if it is not broken down to glucose. Two of the other well-known lysosomal storage diseases would include Hurler syndrome, Hunter syndrome, and the Tay-Sachs disease. Both arise from defects in the degradation of glycosaminoglycan, which are the major carbohydrate components of the extracellular matrix. The defective enzyme in a patient with Hurler syndrome is the alpha-L iduronidase, which is required for the degradation of the glycosaminoglycans. Electron microscopic observation of sweat glands from a patient with Hurler syndrome actually reveals that there's a large number of atypical vacuoles that stain for both acid phosphatase as well as undigested glycosaminoglycans. These vacuoles are apparently abnormal late endosomes that are filled with the indigestible material. Mental retardation is a common feature of lysosomal storage diseases. It can occur due to the impaired metabolism of glycolipids, which is an important component of the brain tissue and the sheets of the nerve cell axons. A very good example of this would be the Tay-Sachs disease, which is a condition inherited as a recessive trait. Afflicted children would show slow mental retardation after about 6 months of age, followed by paralysis and death within 3 years. The disease results from the accumulation in the nervous tissue of a particular glycolipid called the ganglioside. The missing lysosomal enzyme in this case is the beta-N acetylhexoaminidase, which is responsible for cleaving or breaking the terminal N-acetylgalactosamine from the carbohydrate portion of the ganglioside. Lysosomes from children afflicted with Tay-Sachs disease are filled with membrane fragments that contains undigested gangliosides. There are several possible treatments for lysosomal storage diseases, and some can actually be diagnosed prenatally. It is hoped that the treatments that replace the missing enzyme will be developed. This is what you call as enzyme replacement therapy. Another option would be altering the DNA of the patients, called gene therapy, which is another possible approach. Let us now look into the peroxisomes. Like the Golgi complex, endosomes, and the lysosomes, the peroxisomes are bounded by a single membrane. However, they are not derived from the endoplasmic reticulum and are therefore not part of the endomembrane system that includes the other organelles that are discussed earlier. Peroxisomes are found in all eukaryotic cells but are especially prominent in mammalian kidney and in the liver cells. In algae, they are also found as well as in the photosynthetic cells of plants. Peroxisomes are somewhat smaller than mitochondria, though there is considerable variation in size depending on the function and the tissue where they are found. However, regardless of the location or the size, the defining characteristic of peroxisome is the presence of the catalase. This is an enzyme that is important for the degradation of hydrogen peroxide. Hydrogen peroxide is a potentially toxic compound that is formed in several oxidative reactions that are catalyzed by oxidases. Both catalase and the oxidases are confined to peroxisomes. Thus, the generation as well as the degradation of hydrogen peroxide occur within the same organelle. Thus, this protects other parts of the cell from exposure to this harmful compound. This figure shows the animal peroxisome having a distinct crystalline core, which usually consists of a crystalline form of urate oxidase. The crystalline cores contain catalase, which is present in peroxisomes also in the plant's leaves. When such crystals are present, it is easy to identify peroxisome because urate oxidase and catalase are two of the enzymes by which peroxisomes are defined. However, in the absence of the crystalline core, it is not always easy to spot peroxisomes ultracellularly. This figure is an electron micrograph that shows several peroxisomes, or microbodies, in the cytoplasm of a rat liver cell. Peroxisomes occur widely in animals, plants, algae, and some fungi. In animals, peroxisomes are most prominent in liver and in the kidney tissue. There are at least 5 general categories of peroxisomal functions. First would be hydrogen peroxide metabolism, detoxification of harmful compounds, oxidation of fatty acids, metabolism of nitrogen-containing compounds, and catabolism of unusual substances. Hydrogen peroxide metabolism is possible with the help of the enzyme oxidases and also catalases. The oxidases are responsible for generating hydrogen peroxides in peroxisome. On the other hand, hydrogen peroxide is the catalase can also function as a peroxidase. As a peroxidase, the catalase can use a variety of toxic substances as electron donors. This could include methanol, ethanol, formic acid, formaldehyde, nitrates, and phenols. Because all of these compounds are harmful to cells, their oxidative detoxification by the catalase may be a vital peroxisomal function. The prominent peroxisomes of the liver and kidneys are thought to be important in such detoxification reactions. In addition, peroxisomal enzymes are important in the detoxification of reactive oxygen species, such as superoxide anions, hydroxyl radicals. These ROS can be formed in the presence of molecular oxygen during normal cellular metabolism. And if they accumulate, the cell can suffer from oxidative stress. Peroxisomal enzymes, therefore, like the superoxide dismutase, the catalase, and peroxidases, detoxify these reactive oxygen species, preventing them from accumulating and subsequent oxidative damage to cellular components. Peroxisomes found in animals, plant, and fungal cells also contain enzymes that are necessary for oxidizing fatty acids via beta oxidation in order to provide energy for the cell. In fact, around 25 to 50% of fatty acid oxidation in animal tissues happen in the peroxisomes, while the remainder are localized in the mitochondria. In animals, peroxisomal beta oxidation is especially important for degrading long-chain, very long-chain, and even the branched fatty acids. The primary product of beta oxidation, like acetyl coenzyme A, is then exported to the cytosol, where it enters biosynthetic pathways or the tricarboxylic acid cycle. Now, once the fatty acid is shortened to fewer than 16 carbons, further oxidation usually happens in the mitochondria. Thus, in animal cells, the peroxisome is important for shortening fatty acids in preparation for the next metabolic activity that will happen in the mitochondria. Except for primates, most animals require urate oxidase, also called uricase, to oxidize urate. Like other oxidases, urate oxidase catalyzes the direct transfer of hydrogen ions from the substrate going to the molecular oxygen, thereby generating hydrogen peroxide. Additional peroxisomal enzymes involved in nitrogen metabolism include aminotransferases. Members of this collection of enzymes would catalyze the transfer of amino groups from amino acid to alpha keto acids. And these enzymes play an important role in the biosynthesis and degradation of amino acids simply by moving the amino groups from one molecule to another. Some of the substrate for peroxisomal oxidases are rare compounds for which the cell has no other degradative pathway. Such compounds would include the D-amino acids, which are not recognized by enzymes capable of degrading the L-amino acids found in polypeptides. In some cells, the peroxisomes also contain enzymes that break down unusual substances called xenobiotics, which are chemical compounds foreign to biological organisms. This category would include the alkanes, short-chain hydrocarbon compounds found in oil, and other petroleum products. Fungi containing enzymes capable of metabolizing such xenobiotics may turn out eventually to be useful for cleaning up oil spills that would otherwise contaminate the environment. Considering the variety of metabolic pathways found in peroxisomes, it is not surprising that a large number of disorders arise from defective peroxisomal proteins. The most common peroxisomal disorder is the X-linked adrenoleukodystrophy. This defective protein causes the disorder is actually an integral membrane protein that may be responsible for transporting very long-chain fatty acids into the peroxisome for beta oxidation. Accumulation of these long-chain fatty acids in the body fluids would destroy the myelin sheath in the nervous tissues. This ends our discussion on the last part of the endomembrane system. So far, we were able to talk about the roles of the ER and the Golgi complex in protein trafficking, exocytosis and endocytosis, coated vesicles in cellular transport processes. We were able to talk about lysosomes and cellular digestion. And lastly, we ended up our discussion with the peroxisomes, which we said is not a part of the endomembrane system.